Hello friends and welcome. Today I am going to deliver a lecture on measurement of drug effects, kinetic analysis of ligand receptor interactions using Scatchard plot, double reciprocal plot and Hill plot. Let us first try to have a look at the main objectives of today's deliberation. Number first is to give a general and thorough idea about drug action. Number second, we shall be explaining in detail the kinetic analysis of ligand receptor interactions using Scatchard plot. Number third, to discuss kinetic analysis of ligand receptor interactions using double reciprocal plot. And finally, we shall be discussing the kinetic analysis of ligand receptor interactions using Hill plot. Now, we will start with the introduction about drug action. The action of drugs on the human body is called pharmacodynamics, and what the body does with the drug is called pharmacokinetics. The drugs that enter the human tend to stimulate certain receptors, ion channels, act on enzymes or transporter proteins. As a result, they cause the human body to react in a specific way. There are two different types of drugs. Number first is the agonists. That is, they stimulate and activate the receptors, and number second are the antagonists. They stop the agonists from stimulating the receptors. Once the receptors are activated, they either trigger a particular response directly on the body or they trigger the release of hormones and other endogenous drugs in the body to stimulate a particular response. Drugs interact with receptors by bonding at specific binding sites. Most receptors are made up of proteins, and the drugs can therefore interact with the amino acids to change the conformation of the receptor proteins. These interactions are very basic, just like that of other chemical bondings. Number first are the ionic bonds. Ionic bonds occur mainly through attractions between opposite charges, for example, between protonated amino or quaternary ammonium and the dissociated carboxylic acid group. Similarly, the dissociated carboxylic acid group on the drug can bind with amino groups on the receptor. This type of bond is very strong and varies with the inverse of the distance between the atoms, so that it can act over large distances. Cation pi interactions can also be classified as ionic bonding. This type of interaction occurs when a cation, for example acetylcholine, interacts with the negative pi bonds on an aromatic group of the receptor. Ion dipole and dipole-dipole bonds have similar interactions, but are more complicated and are weaker than ionic bonds. Number second are the hydrogen bonds. There is a small but significant attraction between hydrogen atoms and polar functional groups. These so-called hydrogen bonds only act over short distances and are dependent on the correct alignment between functional groups. Receptors are located on all cells in the body. The same receptor can be located on different organs and even on different types of tissues. There are also different subtypes of receptor which elicit different effects in the response to the same agonist. For example, there are two types of histamine receptor, H1 and H2. Activation of the H1 subtype receptor causes contraction of smooth muscle, whereas activation of the H2 receptor stimulates gastric secretion. It is this phenomenon that gives rise to drug specificity. Of course, drugs do not only act on receptors, they also act on ion channels, enzymes and cell transporter proteins. When talking about the shape of molecules, biochemists are mainly concerned with the three-dimensional conformation of drug molecules. There are many isomers of a particular drug, and each one will have its own effects. Differences in isomer affect not only what the drug activates, but also changes the potency of each drug. Potency is a measure of how much a drug is required in order to produce a particular effect. Therefore, only a small dosage of a high potency drug is required to induce a large response. The other terms used to measure the ability of a drug to trigger a response are: number first, intrinsic activity, which defines agonists as having intrinsic activity equals to 1. Number second, antagonists as having intrinsic activity equal to 0 and partial agonist as having intrinsic activity between 0 and 1. Intrinsic efficacy also measures the different activated state of receptors and the ability for a drug to cause maximum response without having to bind to all the receptors. Now, let us discuss the specificity and affinity of drugs. Drug companies invest a significant effort in designing drugs that interact specifically with particular receptors, since non-specific drugs can cause more side effects. An example is the endogenous drug acetylcholine. Acetylcholine is used by the parasympathetic nervous system to activate muscarinic receptors and by the neuromuscular system to activate nicotinic receptors. However, the compounds muscarine and nicotine can each preferentially interact with one of the two receptor types, allowing them to activate only one of the two systems, while acetylcholine itself would activate both. The specificity of drugs cannot be talked about without mentioning the affinity of the drugs. The affinity is a measure of how tightly a drug binds to the receptor. If the drug does not bind well, then the action of the drug will be shorter and the chance of binding will also be less. This can be measured numerically by using the dissociation constant Kd. The value of Kd is the same as the concentration of drug when 50% of receptors are occupied. The equation can be expressed as Kd is equal to drug into receptor divided by complex. But the value of Kd is also affected by the conformation, bonding and size of the drug and the receptor. The higher the Kd, the lower the affinity of the drug. Now, let us discuss the next part of our today's lecture, that is Scatchard equation. The Scatchard equation is an equation for calculating the affinity constant of a ligand L with a protein P. The Scatchard equation is given by R divided by L is equal to NKa minus RKa, where R is equal to L bound divided by P0 is the ratio of the concentration of bound ligand to total available binding sites, and N is the number of binding sites per protein molecule. Ka is the association or affinity constant from the equation, where Ka is equal to Lp divided by L into P. The Scatchard equation is sometimes referred to as Rosenthal-Scatchard equation. Plotting these data, that is R divided by L versus R, yields the Scatchard plot with the slope Ka and a y-intercept of Nka. Relative binding affinities between two sites can be distinguished with a line showing identical affinity and a curve showing different affinities. The Scatchard equation is named after the former MIT chemistry department member George Scatchard, an American chemist. Next part of our today's lecture is Scatchard plot. A Scatchard plot is a plot of the ratio of concentrations of bound ligand to unbound ligand versus the bound ligand concentration. It is a method for analyzing data for freely reversible ligand receptor binding interactions. The plot yields a straight line of slope k, where k is the affinity constant for ligand binding. The affinity constant is the inverse of the dissociation constant. The intercept on the x-axis is Bmax. It is sometimes the case that binding data does not form a straight line when plotted in a Scatchard plot. Such is the case when ligand bound to substrate is not allowed to achieve equilibrium before the binding is measured or binding is cooperative. In a Scatchard plot, assumptions of independence in linear regression model is violated because B bound ligand is used in the x and y axes. Generally, Scatchard and Lineweaver-Burke plots are outdated. Their original intention was to transform the data into linear representations of the original data such that linear regression methods could be applied. These transformations frequently distort experimental error and can be misleading if results are not accurate. Now, coming to the limitations of Scatchard analysis. First is that R and R divided by L both contain experimental errors. Hence, calculating the slope and intercept of a linear Scatchard plot by simple least squares is not statistically valid. In addition, non-linear Scatchard plots are notoriously difficult to interpret. Another source of overinterpretation stems from the fact that binding behavior consistent with multiple classes of sites is also consistent with other binding models. In the case of multiphasic plots, discerning the true molecular properties of a receptor requires independent experiments in addition to Scatchard analysis. Now, let us discuss the next part of our today's lecture, that is double reciprocal plot. In biochemistry, the Lineweaver-Burke plot or double reciprocal plot is a graphical representation of the Lineweaver-Burke equation of enzyme kinetics described by Hans Lineweaver and Dean Burke in 1934. The plot provides a useful graphical method for analysis of the Michels-Menten equation, that is V is equal to V max S divided by Km plus S. Taking the reciprocal gives 1 divided by V is equal to Km plus S divided by V max into S is equal to Km divided by V max into 1 divided by S plus 1 divided by V max, where V is the reaction velocity, that is the reaction rate, Km is the Michels-Menten constant, V max is the maximum reaction velocity, and S is the substrate concentration. Now, coming to its uses. The Lineweaver-Burke plot was widely used to determine important terms in enzyme kinetics such as Km and V max before the wide availability of powerful computers and non-linear regression software. The y-intercept of such a graph is equivalent to the inverse of V max. The x-intercept of the graph represents -1 divided by Km. It also gives a quick visual impression of the different forms of enzyme inhibition. The double reciprocal plot distorts the error structure of the data, and it is therefore unreliable for the determination of enzyme kinetic parameters. Although it is still used for representation of kinetic data, non-linear regression or alternative linear forms of the Michels-Menten equation such as the Haynes-Wolff plot are generally used for the calculation of parameters. When used for determining the type of enzyme inhibition, the Lineweaver-Burke plot can distinguish competitive, non-competitive and uncompetitive inhibitors. Competitive inhibitors have the same y-intercept as uninhibited enzyme, but there are different slopes and x-intercepts between the two data sets. Non-competitive inhibition produces plots with the same x-intercept as uninhibited enzyme, that is Km is unaffected, but different slopes and y-intercepts. Uncompetitive inhibition causes different intercepts on both the y and x axes. Now, coming to the disadvantages of double reciprocal plots. Because they involve grossly uneven weightings of points, the linear and formally similar double reciprocal Benesi-Hildebrand and Lineweaver-Burke plots should never be used to resolve equilibrium and enzyme kinetic results. In such plots, weightings from one end to the other may vary as much as 1,000 or more. More equal weightings of the experimental points may be obtained by utilizing the linear single reciprocal Scott and Haynes plots. The most appropriate resolution is achieved by a non-linear least squares analysis with the most error-prone observable expressed as a function of all other variables and parameters. Now, let us discuss the last part of our today's lecture, that is Hild plot. Molecular binding is an interaction between molecules that results in a stable physical association between those molecules. Cooperative binding occurs in binding systems containing more than one type or species of molecules and in which one of the partners is not monovalent and can bind more than one molecule of the other species. For example, consider a system where one molecule of species A can bind two molecules of species B. Species A is called the receptor and species B is called the ligand. Binding can be considered cooperative if the binding of the first molecule of B to A changes the binding affinity of the second B molecule, making it more or less likely to bind. In other words, the binding of B molecules to the different sites on A do not constitute mutually independent events. Cooperativity can be positive or negative. Cooperative binding is observed in many biopolymers including proteins and nucleic acids. Cooperative binding has been shown to be the mechanism underlying a large range of biochemical and physiological processes. The first description of cooperative binding to a multi-site protein was developed by A.V. Hill. Drawing on observations of oxygen binding to hemoglobin and the idea that cooperativity arose from the aggregation of hemoglobin molecules, each one binding one oxygen molecule. In biochemistry and pharmacology, the binding of a ligand to a macromolecule is often enhanced if there are already other ligands present on the same macromolecule. It describes the fraction of the macromolecule saturated by ligand as a function of the ligand concentration. It is used in determining the degree of cooperativeness of the ligand binding to the enzyme or receptor. A coefficient of 1 indicates completely independent binding regardless of how many additional ligands are already bound. Numbers greater than 1 indicate positive cooperativity, while numbers less than 1 indicate negative cooperativity. The Hill coefficient of oxygen binding to hemoglobin is 2.3 to 3.0. The Hill equation can be described as theta is equal to L raised power n divided by Kd plus L raised power n is equal to L raised power n divided by Ka whole raised power n plus L raised power n is equal to 1 divided by Ka raised power n divided by L plus 1, where theta is the fraction of the ligand binding sites on the receptor protein which are occupied by the ligand. L is the free unbound ligand concentration. Kd is the apparent dissociation constant derived from the law of mass action, that is equilibrium constant for dissociation. Ka is the ligand concentration producing half occupation, that is ligand concentration occupying half of the binding sites. N is the Hill coefficient describing cooperativity or possibly other biochemical properties depending on the context in which the Hill equation is being used. Taking the reciprocal of both sides, rearranging, inverting again and then taking the logarithm on both sides of the equation leads to an alternative formulation of the Hill equation, which is represented as log theta divided by 1 minus theta is equal to n log L minus n log Ka is equal to n log L minus log Kd. When appropriate, the value of the Hill coefficient describes the cooperativity of ligand binding in the following way. N greater than 1 means positively cooperative binding. Once one ligand molecule is bound to the enzyme, its affinity for other ligand molecules increases. N less than 1 means negatively cooperative binding. Once one ligand molecule is bound to the enzyme, its affinity for other ligand molecules decreases. And n is equal to 1 is a non-cooperative binding. The affinity of the enzyme for a ligand molecule is not dependent on whether or not other ligand molecules are already bound. The Hill plot is obtained by plotting log theta divided by 1 minus theta versus log L. In the case of the Hill equation, it is a line with slope nh and intercept log Kd. This means that cooperativity is assumed to be fixed, that is, it does not change with saturation. It also means that binding sites always exhibit the same affinity and cooperativity does not arise from an affinity increasing with ligand concentration. With this, we come to the end of today's lecture. Hope you have understood it very well. Thank you very much and goodbye.